Hi, I'm Candice Ng.
Hello, I'm Aaron Huang.
Hi, I'm Alvin Zhang.
Hi, I'm Flora Zhang.
I'm Savini Ho from Lowell Team C.
Cancer is the collection of related diseases caused by uncontrolled cell proliferation. There are many types of treatments for cancer, including surgery, radiation therapy, chemotherapy, and also stem cell transplants. And researchers have been also talking about using T cells for cancer therapy. However, there is a problem by using T cells because the cancer cells do not display certain antigens that the T cells need to recognize. Thus, cancer remains a fatal disease resulting in tumors, which cause high percentages of deaths.
T cells play a crucial part in your body's immune system to fight foreign invaders such as bacteria, viruses, or parasites. T cells also have the ability to fight cancer with the use of CAR, or chimeric antigen receptor. A CAR is any antigen that has been genetically engineered to be placed in a T cell. In our project, we plan on inserting carcinoembryonic antigen into the T cell by using viruses as vectors because they can deliver the gene by infecting the T cell. We will then use a chemical switch called rapamycin to activate the CAR T cells once they are inside the body. Since many T cells do not present their antigens on the surface of the cell, we plan on using radiotherapy to upregulate MHC1. This will allow interior antigens to be presented to the outside where our CAR T cells can bind to the antigen and destroy the tumor cell.
There's another major problem that we need to solve. Normally, CAR T cells cannot enter solid tumors and become exhausted on the way in. Therefore, we will use a virus to edit the genes of the CAR T cell, specifically removing the PD-1 program death 1 pathway responsible for cell exhaustion. This process is similar to that of creating the CAR T cells from T cells and has been tried, tested, and used to treat lymphoma successfully. We will replace it with genes from another pathway, which causes the CAR T cells to only become active in the presence of the drug rapamycin. This prevents the CAR T cell from becoming cancerous itself. The transformed CAR T cell, which only is responsive in the presence of the drug, is now called the Go CAR T cell because of its response, and can now successfully destroy the tumor cells.
To test the efficacy of our treatment, we will first test it on mice before holding trials on humans for obvious safety reasons. The trials with mice will review what type of side effects we need to take into account for how to modify and improve our process moving forward. We will use a group of genetically identical mice to eliminate any possible error due to random genetic variability. There will be a control group induced with colon and breast cancer but with no treatment, and another experimental group exposed to the same types of cancer with our treatment. We are aware that not all mouse models can mimic the same diversities and symptoms found in human breast cancer, but we can manipulate one of the strains of the mice to mimic a specific type of human breast cancer that will be kept consistent throughout both groups. To replicate our treatment for mice, we will extract T cells simultaneously from their thymus and blood. After attaching a similar chimeric antigen receptor to it along with the rapamycin and expand it in the laboratory, we will inject back into the located target of the mice. Screening of the cancer cells in mice will be made weekly. We will compare the groups of mice to the efficacy of other traditional cancer treatments in addition to the control group of mice with no treatment at all. For our treatment to be considered effective, over 80% of the experimental group must show a reduction of cancer cells compared to the control group, and 50% of the tumors in those who show reduction must be at least 50% less than the control group, while also not demonstrating any serious, long-lasting side effects. Some ethical and societal concerns we have to consider would be how we manipulate the mice. To inject cancer in mice or any animals for experimental purposes has always been an ethical controversy. Next, in the case that we move to the phase of clinical procedures, we will need to have precautions to deal with potential side effects, one of which is how the injection of T cells will cause an abnormal increase of cytokines, which will directly lead to an immune response, which may keep patients from being safe in the procedure of testing the treatment. To keep the patient safe will be something we must consider. We are also not fully aware of the powers of the GO CAR T cells and its potential behavior of attacking neighboring healthy cells.